So I'm going to talk a bit about some of the work that we've been doing over the last 10, 15 years or so. But to begin with, I'm going to confess a bit of a panic. So when I was preparing this talk, I realized I didn't really have a great definition in my head of what an animal mind was. I'm not a psychologist by background, so I'm not necessarily used to some of the literature in this field. So I was kind of relieved when I started doing some reading and found this definition by Minsky, who said that minds are simply what brains do. So I was kind of relieved by that because I can relate to that at least. It's an interesting essay. I think Minsky was an early pioneer of AI, and he was discussing his idea of what minds are. And he had this nice passage which he said, we can envision the mind like a great empire of countries, towns, and villages. Each part has strong connections to certain neighbors, but much weaker connections with most of those that lie further away. Each of them can learn to influence some of the others, and thus to exploit the skills of others that those others learn. And to understand those vast societies of mind, which was the title of the book that he was plugging, inside ourselves, we will have to develop good ideas about how mental phenomenon emerge from communities of simpler systems. And I like this passage because it kind of chimes with what we're trying to do in the system that we're working on, which is to try and understand how a neural circuit has evolved to facilitate a behavioral innovation. And it fits with kind of what I wanted to talk about anyway, which is that by studying brains and behavior in parallel, you can make inferences about adaptive processes that are maybe a bit more precise than you can in isolation. And obviously, hopefully one day, those two things will line up and give you an indication of causation. And as a comparative biologist, also favor the idea that natural variation in behavior gives you an in the sense that they provide variation that you can use to understand behavior. So the system that we mostly work on are a group of tropical butterflies called Heliconius, and they're unique among butterflies as the only butterflies that feed on pollen. So most butterflies you'll be familiar with are nectar feeders. So they ingest most of their protein during the larval stage, and then they build up protein stores as a caterpillar, and then their adult stage is basically using up these protein stores, and when they're done, they die, basically. So they run out of protein to produce eggs and sperm, so they lose their reproductive fitness, and there's narrow selection to maintain their longevity. What we can see in this photo is that Heliconius are capable of collecting pollen grains on their proboscis. They don't have any mandibles, so they're not able to crunch these things up. So what they seem to do is to secrete some sort of enzymatic solution onto the proboscis. They then unwind their proboscis over and over again, and that seems to break up the pollen grains, and that releases the amino acids in the pollen into solution that they can then suck up. And this has had a really big impact on their life history, so they can live for up to 6 months, which is 3 or 4 times longer than their closest relatives. And they can also maintain their reproductive output for that entire time. So they don't show the classic female fecundity curve. They can maintain their reproductive output for their entire lifespan. So it's a big innovation in their behavior and life history, but they do seem to be constrained in a sense that they can only collect pollen from specific flowers. And these flowers seem to occur at low density in tropical forests. So when you're faced with a low-density resource in a complex environment, one of the best strategies you would think would be to learn the location of those resources. And these resources are quite stable, so these plants can flower for up to a year, which is much longer than the lifespan of the butterfly, or mostly. So what was thought that Heliconius do, and what was described in the 70s, is that they have evolved a nocturnal roost, so they return to the same sites, or 1 or 2 sites every night. And then they visit the same floral resources in the same order at the same time of day, kind of like a bus route. So this is very similar to the trapline foraging behavior that you see in bumblebees and some other bees. And it's based, or it's thought to be based on visually orientated spatial memory. So soon after this was described, it was pointed out that it must require some sort of shift in the nervous system to produce these behavioral changes. And we have a good idea of the circuits that must be involved in this behavior from comparisons with other insects. And these involve sensory integration centers called the mushroom body and the central complex. So this is a cartoon of a butterfly brain, and it describes the sort of general circuits that we're thinking about. So either side of the central brain, which is here, we have the visual centers, so these are the optic lobes. So they're responsible for collecting visual information that's then fed forward into the mushroom bodies and other parts of the central brain. Similarly, you have the antennal lobes in blue here, which collect or process olfactory information, and they also send that downstream to the mushroom bodies. So in the mushroom bodies, you then have integration of different sensory modalities, internal cues, and past experience. So the mushroom bodies are the center of learning and memory in insects. The central complex is then involved in directing behavior in a number of different ways. So it's linked to navigation, which is critical for traplining behavior. So when we've been thinking about this general circuit, the things that we're interested in is how you might change this general flow of information to produce traplining behavior from ancestor that was foraging more stochastically. And there are a number of ways that you can think that you might be able to do this. So you can innovate new cell types that might process information in a different way. This could happen in the periphery, so in the visual centers or in the mushroom body itself. You could also change the way that you integrate different types of information, so combining different sensory cues. Or you could change the way that the brain is processing the meaning of that information, so changing the valence. So ostensibly, you could, in theory, imagine lots of different ways to change the circuit to produce something like traplining behavior. But we also believe that there are sort of constraints acting on this system that limit the sort of adaptive potential. And that's kind of what we see, because what we look at, or what we see when we look at the neuroanatomy are very specific changes in brain morphology. And in particular, we see really big changes in the mushroom body, but very little changes anywhere else. So this is a 3D model of a Heliconius brain from the front and the back, and the mushroom bodies are now in red. So we have the anterior structure called the lobes, and the posterior structure called the calyx. And these are formed of a group of cell types called Kenyan cells. This is just a cross-section of an antibody-stained image through the lobes and the calyx in a Heliconius, so a pollen-feeding butterfly. And this is a comparison with one of their closest relatives, a Growlus vanille, which lives in the same habitats, exploits the same host plants, but doesn't pollen feed. And hopefully you can easily see that there's a really big investment difference in the mushroom body. So one of the things that we've been trying to do is to map out this variation in a lot more detail. So I spent a long time traveling around South America trying to collect as many different species of Heliconius and outgroups as possible. So in red here you have the Heliconius themselves. There's about 50 species. I think we managed to get about 30 of them. You then have a series of other genera which are less speciose. So you have the Uades, which is the closest relative to Heliconius, and a few other genera. And these are fairly closely related. So the last common ancestor of Heliconini was about 20 million years ago. Last common ancestor of Heliconius was about 12 million years ago. And what we've been trying to do is map out variation in mushroom body size. And one of the easiest ways you can do that is just to plot log mushroom body volume against the rest of the central brain as an allometric control. And this is kind of what you see. So first of all, you see a lot of variation up the y-axis. So from the smallest to the largest mushroom body, we see a 25-fold difference in investment. If you look at these scaling relationships, these are estimated scaling shifts between different parts of the phylogeny. So that reflects the colors on these branches. And what it tells us is that these sort of early branching lineages, so the other Heliconini genera, have fairly low investment in mushroom body size relative to the rest of their brain. And then you see a series of independent shifts across the phylogeny where mushroom bodies expanded, but the rest of the brain hasn't. So these are these yellow dots. The main thing I want you to focus on is this sequence, so going from the blue line to the purple line to the red line. Each time you progress up those lines, you're doubling mushroom body size relative to the central brain. And so these occur in sequence in the origin of Heliconius, which are these red data points. So when you compare Heliconius to these early branching lineages, you have a fourfold increase in mushroom body size relative to the central brain. We've also counted the number of Kenyan cells, so these are the number of intrinsic neurons in the mushroom body. And this fourfold increase is actually under reflecting the change in the neuron number, so we actually see an eightfold increase from something like a Dryas to a Heliconius. So this is a really big shift in brain morphology over a fairly recent evolutionary period. And the interesting thing is if you look at other parts of that circuit that I described, you don't see anything obvious that matches that in any way. So if you look at the olfactory pathway, we've been able to map out the main tracks that indicate the flow of information from the antennal lobe to the central brain. And they're very similar between the outgroups and Heliconius. And if you look at investment in the main olfactory neuropile, the antennal lobe, you don't see any change between Heliconius and the outgroups. So Heliconius here in red, everything else are the outgroups. And similarly, if you look at the visual pathway, which is perhaps more interesting here, we can identify all the main tracks in each of these genera, and we find the same things. The morphology is slightly different because the brain is slightly deeper, but the tracks are there, and everything seems to be pretty conserved. And again, if you look at investment in the visual neuropile, this is just picking out the medulla, which is the largest visual neuropile, you see really consistent scaling relationships between Heliconius and everything else. Similarly, if we look at some of the downstream circuits, like the central complex, we don't see any obvious change there. So it seems, in terms of overall brain morphology, there's a very specific shift in brain composition that is accompanied by the evolution of pollen feeding. But things get a bit more interesting when you start to look within the mushroom body itself. So while we don't see specialization in the sensory circuits themselves, within the mushroom body, we do see evidence of visual specialization. And the way that we can detect this is to inject different colored dyes into the olfactory and visual pathways, and then see how these dyes migrate along those tracks to the mushroom body itself. And this delineates different areas of the mushroom body calyx, which is the posterior region that processes visual and olfactory information. So this, for example, is a background stain showing the outline of the calyx. This is an injection into the antennal lobe, so this green area is the calyx that processes olfactory information. This is the same thing for the visual pathway, so the magenta area is the area that processes visual information, and this is these two stuck together, so you can see a segregation of the olfactory and visual calyx. This is just an excuse to show a pretty video. This is a Z scan through the depth of one of these calyxes that shows that these areas are segregated throughout the whole structure. So that's kind of handy as well, because we can then estimate the volume of the visual and olfactory calyx. We've done this in a group of different species, so 4 Heliconius and 4 of the outgroups. And then again, we can look for consistency or lack of consistency in scaling by plotting the visual calyx against the olfactory calyx. And when we do that, we find, again, one of these big grade shifts up the y-axis. So these are all of the outgroups that tend to scale in a fairly consistent way. There's some variation, but it's more or less consistent. And then the red line are the Heliconius. And again, if you look up the x-axis, you intercept these two lines at different points, which implies that for any given volume of olfactory calyx, Heliconius have twice as much visual calyx. So we infer that this is sort of an indication that Heliconius mushroom bodies are specialized to process visual information, or at least they've increased the amount of processing power that they're dedicating to processing visual information. That's the posterior end of the mushroom body, but you also see interesting things happen at the other end of the mushroom body, at the anterior side, where the mushroom body lobes form. So I've mentioned these Kenyan cells that comprise the mushroom body. And they're a particular cell type, but within the Kenyan cells, you have different subtypes. So you have different categories of Kenyan cells, which we know in other insects are linked to specific types of learning and memory. In Heliconius, we don't know much about those cell types, but what you can do is take cross-sections through the peduncle, which is the bundle of axons that connects the calyx to the lobes. And through various staining procedures, you can identify bundles of tracks which reflect these different Kenyan cell subgroups. So we can identify different compartments of these tracks that would correspond to different Kenyan cell subtypes. If we do this in Dryas, which is one of the small mushroom body species, and a Heliconius that has a big mushroom body, you can see just from the cross-sections alone that there's some differential shift in the sort of proportions of these things. But if you then follow these tracks out into the lobes themselves, where they expand out, you can produce these 3D models of different substructures within the lobes. And those different lobes seem to be overall quite well expanded in Heliconius, but we also see some evidence that some are differentially expanded relative to others. So we can take this volumetric information, do a principal component, and you can see the major axis of variation is along one principal component that reflects overall size. We also see that there are some loadings that are going in different directions that indicate something else is going on. And through various analyses, we can sort of pin this down to kind of two groups of lobes. One which comprises alpha, beta prime, and gamma, and one that's alpha prime and the Y. And if you compare investment in these two categories of lobe structures, you again see that the outgroups in blue invest much less in this group of lobes relative to Heliconius. So even within the mushroom body circuit, there's been specific expansions of some subcomponents of cell types over others. And in this case, again, looking at homology with Drosophila in particular, these are the lobes that are involved in processing or forming long-term memories. So we think this again gives us a clue about behavior. Just as a final note, we've also been able to look at some of the other cell types that interact with the mushroom body. So these are neurotransmitting modulatory neurons that interact with the mushroom body to either change how that information in the mushroom body is being interpreted in terms of valence, or also feeding forward to other structures in the brain. And again, the overall picture is that there's largely conservation, so we can identify these different clusters of cell bodies which have similar patterns of tracks connecting to the mushroom body and express similar neurotransmitters. So overall, the general circuit is well conserved, but if you then look at specific cell types reflected by these cell groups, you can see some interesting patterns emerge. So in general, again, most of the cell numbers are conserved, but there's a particular group, which are the GABAergic feedback neurons, which we think have doubled in number between Heliconius and the outgroups. And these feedback neurons have an interesting structure in that they project to both the calyx and the lobes. And in Drosophila, they're shown to be important for ensuring sparse coding of information. So sparse coding is just the idea that you have different Kenyan cells that respond to very specific combinations of cues, which means that stimulus is reflected in quite a unique way within the mushroom body. And what that allows you to do is have a particular combination of responsive neurons for a certain cue and a different combination for a related cue, which gives you precision in comparing those stimuli. So we think this difference in cell numbers in these feedback neurons reflects a potential shift in the precision of visual processing within the mushroom body. So that was quite a lot of information, but I'm just going to summarize it very quickly before moving on to some behavior. But what I think we see when we look at our system, at least, is that you have very targeted shifts in the way that information is being used in the brain rather than the way that information is being captured. You also see sensory specialisation within the mushroom body that indicates that specific sensory contexts are important for this group of butterflies. And within the mushroom bodies, you also see expansion of specific cell types. We think that these are probably related to specific memory profiles or specific learning profiles. And the overall commonality of this kind of information is that you see very localized changes. So these are not whole suite changes that are affecting the whole brain or even the whole mushroom body. These are very specific shifts in the way that this circuit is functioning that indicate specialization towards these behaviors. So given that's the case, you would expect to see a degree of specificity in the sort of cognitive changes between Heliconius and their relatives. And that's what I'm going to talk about for the rest of the talk. Obviously, when we're talking about pollen feeding and trap lining behavior, the main behavior that you would think of is spatial learning and spatial memory. And we have done these kind of experiments. So this is a setup that we had in a facility in France where we could build these very large T mazes. So there's a series of connected cages that each were 10 by 10 meters in size. And they were connected by these corridors of cage that were also 10, 15 meters long. So the total width of the T maze is almost 60 meters, and you can train Heliconius to preferentially forage in one arm or the other. So we can show that they do indeed learn spatial information. We haven't been able to do this comparatively for a couple of different reasons. But the other approach that we've kind of taken is to expose Heliconius and their relatives to a kind of battery of different cognitive tests. So we've looked at a variety of things like sight fidelity, so the ability to learn and maintain a home range, long-term memory, reversal learning, circadian memory, non-elemental learning, shape learning, social learning, landmark learning, and spatial learning. And for all of these experiments, the general idea is that we ask two main questions. So first of all, we ask whether or not Heliconius can solve the tasks that we give them. And then we ask whether or not Heliconius can solve them better than the outgroup Heliconini that have smaller mushroom bodies. And in general, the answer is that Heliconius can solve most of these tasks, but they're not usually much better than the outgroup genera. So there's only two experiments that we've done where we found a significant difference between the performance of Heliconius and the performance of their closest relatives that have much smaller mushroom bodies. So these are in long-term visual memory and non-elemental learning. So what we think this sort of indicates is that there are sort of specific cognitive toolkits that are used in the evolution of pollen feeding in Heliconius to produce these trap lining behaviors. So I'm going to explain these two tests in a bit more detail. So non-elemental learning is interesting because it's trying to get at how the brain processes patterns of information. So rather than associating one cue with a positive or negative reward, it's associating combinations of cues. So the sort of level 1 non-elemental learning is what's called positive pattern learning. So in this case, you have a series of artificial feeders, purple and yellow, and then the combination of purple and yellow. And you positively reinforce the combination, so the purple and yellow flower, and negatively reinforce the pure color flowers. And we've done this in Dryas Julia, which is one of the smaller mushroom-bodied outgroups, and in Heliconius. And what these graphs all show is the proportion of correct responses, so in this case, the proportion that goes to the purple and yellow feeder, for naive butterflies, and then the same butterflies after a training period, so in this case, a 4-day training period. And each dot here is an individual. So what you can see is that both of these species respond to training in that they shift their preference up the y-axis, but Heliconius are actually much more accurate in this ability, and that's a significant species effect. You can also then make this test harder by going to sort of level 2 non-elemental learning, where you introduce more complex combinations of cues. So in this case, we've got 4 different colors, yellow, purple, red, and blue, and you positively reinforce a certain combination of those cues and negatively reinforce another combination. So in this case, for example, yellow and purple are positively rewarded, blue and red are positively rewarded, but blue and purple and yellow and red are negatively rewarded. So color contains no useful information in this sense. The main source of informative stimuli is the combination of the colors. And when we get Dryas to do this test, they are not very good at it. They don't show a significant shift in their preference after training, whereas Heliconius do seem to be able to do it. The sample size here is slightly lower because we were halfway through this experiment in 2020, so that's a COVID excuse. But that does seem to be the case, at least. And that's, again, a significant species effect. The other case where we find this effect that Heliconius have an improved performance over the outgroup, so when we look at long-term visual learning. So this is a much longer behavioral experiment. It takes at least 3 weeks. But we start off with freshly closed butterflies. They have a few days feeding off white feeders just to get them used to the feeder setup. We then give them a naive preference test, so we ask them if they have a preference between 2 different colors, like purple and yellow. And then we train them to positively associate one of those colors with a reward, so in this case purple, whilst yellow is negatively rewarded. Give them a training period of 8 days. We then do a recall test to check whether or not they've learned that association, so again with empty feeders. And then we give them white feeders again for another 8 days. So this is sort of a neutral stimulus relative to the learning conditions. And then at the end of those 8 days, we test their preference for the red and the purple and yellow again. So the idea is to see whether or not they remember an induced preference over a long period of time. And obviously you can repeat this sequentially, so you can test their preference at 18 days and then give them another 8 days with the white feeders and see how long they can maintain their association. In this case, we've done it across 6 species, so 3 Heliconius and 3 of the outgroup genera. And this is kind of what the results look like. So again, you've got the proportion of correct choices up the Y. We have the untrained preference in the white boxes. This tends to be less than 50-50 because we train them against their naive preference. You've got the trained preference, which is usually between 90 and 100%. And then you've got the long-term memory recall, which is after that 8-day period with the white flowers. So in this case, Heliconius aurata has maintained their learned association pretty accurately. If you look across the 6 species, you see more or less the same pattern, but there's a kind of trend towards decreasing long-term memory performance in the outgroups. So if you separate them into the Heliconius and the outgroups and then look at their sort of mean long-term memory performance, you can sort of see that there's this shift where Heliconius maintain their learned associations much more faithfully than the outgroups do. That's a significant difference, and for various reasons, we've repeated this experiment about 4 times, much to the disdain of my lab. But it's always really robust, and so it seems to be a very clean result. One thing that we've done recently is to repeat the experiment, but now instead of using visual cues, we use olfactory cues. So the idea was to test whether or not you have any evidence of modality-specific memory enhancement. So this relates both to their sort of natural behavior in the sense that we think that they learn these trap lines using visual cues rather than olfactory cues, which are less reliable in the environments that they live in, and also the neuroanatomy, where we see evidence of visual specialisation in the mushroom body. So we've done these experiments across 2 Heliconius and 2 outgroups now, and we basically try and make the experiment as similar as possible to the visual learning experiment, but using odors instead of colours. And obviously, to do that, we have to separate the 2 sort of training sets in space so that the odors don't mix. But that's more or less the only difference in the setup. In this case, we again have naive preference, recall, and the long-term memory performance. And now what you'll hopefully notice is that performance in these long-term memory assays is pretty similar between Heliconius and the outgroup genera. For example, if we compare that to the data for the visual trials from the same species, this is the shift that we see in the visual test, whereas we don't see that in the olfactory test. And if you combine the data for the 2 Heliconius and the 2 outgroups together, you can summarize it in this plot, which shows sort of overall performance in naive, the learnt preference, and then what happens in the long-term memory trial. And you can see for the outgroups, the performance for visual and olfactory long-term memory is pretty similar. But in Heliconius, which are the purple lines there, you get this split in performance where they're much more accurate in their visual long-term memory than their olfactory long-term memory, which again fits the predictions that we had based on their specialisation within the calyx. So to summarize this behavioural stuff, the main thing to take home is that in general, Heliconius and their outgroups are pretty much performing very similarly across most of the assays that we've done. But what we see is very specific enhancements in cognition or learning and memory in Heliconius. So we see this improved precision of cue comparisons in the non-elemental tests. And then we also see this improved long-term memory performance in the long-term memory tests. And more recently, we've also got evidence that these memory enhancements are modality specific, so they occur in one sensory setting, but not the other. So just like we saw in the neuroanatomy studies, again, the main take-home message for me is that there's a lot of specificity in the behavioral shifts that they're showing. So if we think about how selection is acting on behavior, if you combine these sort of behavioral and neuroanatomical data sets, for this system at least, what we see is that there's very specific localized changes in neural circuits. These affect specific memory profiles and specific sensory modalities. So ostensibly, even though what we're talking about is a case where diet or a dietary innovation is shifting behavior of a particular group of species, it's actually much more specific than that in the sense that we are seeing quite targeted changes in behavior and the neural circuits that support those behavior. So I would argue that if we're trying to answer this question, that we really need to be thinking about what information is being used to produce a certain behavior. And the changes that we will observe in both the neural circuits and the behavior will relate to that specific information and the way that that information is processed. So if that information is processed by a specific part of the brain, you would expect to see that shifts in that part of the brain. But if it's distributed across wider neural circuits, then you might have shifts in larger components of brain structure. And the other point that I would make is that these selection pressures might shape information use in different ways, even when you have a source that is nominally similar. So for example, if you have a dietary shift in two different independent lineages, you might see different responses because those lineages might be using information in different ways. That's kind of related to our specific system, which is kind of unique, as I said. But I also sort of, from my own reading, I take home similar messages from other systems. So some of my favorite systems at the moment, thinking about electroreception in mormyrids, facial recognition in paper wasps, and spatial memory in chickadees, some of which we'll hear about later, so I don't want to embarrass myself by misinterpreting these studies. But my general take-home message is that when you look at these systems, you have quite specific shifts in behavior and quite specific shifts in the neuroanatomy that underlie their behaviors. These are kind of similar in a way to Heliconius, where you have major adaptive radiations or variation among closely related populations or species. But I'd also argue that you see similar patterns in the comparative literature as well. So you look at brain structure, for example. So Rob might talk about this later, but one of the classic debates in comparative vertebrate neuroanatomy is the role of diet and sociality in shaping brain size, which is used variably as a proxy of cognition. But one of my sort of favorite examples is when you look at primates, in particular, in both nocturnal and diurnal primates, you see this association between diet and brain size. I'm not saying it's the only association, but it seems to be there. But what Rob showed almost 30 years ago now is that this pattern is at least partially driven by selection on different sensory systems. So in the nocturnal primates, you see associations between components of the olfactory system and diet, which seem to be linked to changes in brain size. Or in the diurnal primates, you see selection acting on the visual system. So even though these are sort of similar categories of ecological variation, the information that these species are using is different in these different contexts, and therefore the response of the brain is also likely to be different. So I'm going to end there. I'm going to thank Antoine, who did a lot of the comparative neuroanatomy, Fletcher Young, who did the behavioral experiments I talked about, Priscilla, who did the spatial learning memory experiments, Max, who did the really tedious lobe reconstructions, which required a lot of effort, and Elizabeth, who's been doing the olfactory long-term memory experiments. But lots of people contributed to this project in different ways, including Owen McMillan, who's our collaborator at STRI, and thanks to Ellie, Alex, and the Royal Society team for this great meeting, which I'm looking forward to now that I've finished my talk.